2 million years ago, South Africa, an early forger is crafting a tool. These carefully selected lithic cores are struck with technique and dexterity. This is an early handyman at work. In the distance, a group of cheetahs is dismembering the remains of an impala. Homo Habilis seeks something to eat, and though this seems like a risky situation, this group is up for the challenge, as it will provide a sizable meal. The group gets closer and closer, until they attempt to confuse the cheetah to grab their portion. The cheetah fights back, unfazed by their actions. However, one of the group grabs part of the carcass and runs quickly to safety. The cheetah is then overpowered by the group, proving the power of collaboration. With their newly made stone tools, they find it easy to butcher meat. The Homo Habilis group consumes high quantities of meat, with scientists suggesting this led to the growth of their brains. As some of the first humans, Homo Habilis was part of a lineage that led to us, modern humans. Africa is perhaps one of the most historically rich sites of early hominin fossils worldwide, containing significant discoveries like Lucy, an Australopithecus afarensis, and the Tong child, a young Australopithecus africanus, that have provided incredible insights into our evolutionary history. Africa, also known as the cradle of humankind, was the home of our species, Homo sapiens, with fossil evidence placing the origin of our species at about 300,000 to 200,000 years ago. Human beings evolved in Africa from a variety of hominid species over a period of several million years. Many hominin species coexisted or overlapped with one another for nearly a million years. To think about how exciting and intriguing it would have been if some other hominin lineages thrived till today, making a world full of diverse hominin species is simply fascinating. We would have a completely different societal structure and unique culture. About 3 million years ago, global cooling brought a steep decline in rainfall in East and Central Africa, where our human ancestor, the Australopithecine, lived. Australopithecus were small, slow, ape-like hominins whose discovery proved bipedality preceded increase in brain size. Forests and woods gave way to savannas and grasslands, and stimulated the evolution of Homo habilis. The discovery of its remains by the Leakeys at the Olduvai Gorge in Tanzania yielded rich insights into the evolutionary history, technology, diet, and anatomy of this intriguing primeval human. Hi, welcome to Enchiridion. I am honored to share with you these facts on Homo habilis as a detailed episode of the series Primeval Human. Homo habilis, meaning handyman, was an ancient human from the Gelasian of the early Pleistocene of East and South Africa about 2.3 to 1.65 million years ago. Similar to contemporary Homo habilis brain size generally varied from 31 to 55 cubic inches, or 500 to 900 cubic centimeters. The body proportions of Homo habilis are only known from two highly fragmentary skeletons, including OH62, and to fill these gaps, paleoanthropologists have estimated sizes based on the anatomy of the earlier australopithecines. The presumed female specimen OH62 is typically estimated as having been 3 feet 3 inches to 3 feet 11 inches, or 100 to 120 centimeters in height, and 44 to 82 pounds, or 20 to 37 kilograms in weight, assuming australopithecine-like proportions. Yet assuming human-like proportions, she would have been about 4 feet 10 inches, or 148 centimeters, and 77 pounds, or 35 kilograms. As postulated for australopithecines, Homo habilis may have been at least partially arboreal. Early hominins are typically reconstructed as possessing thick hair and marked sexual dimorphism, with males much larger than females, though this has led to debate amongst paleoanthropologists. Despite the fact that it was a close human ancestor, it still retained ape and australopithecine-like traits, including a moderately prognathic face, long arms, and an ape-like body, similar to that of the Australopithecine, while also using trees for retreat and protection. Brain case impressions, taken from inside the skull, suggest that Homo habilis showed development in areas of the brain pertaining to language, indicating a language or proto-language. Homo habilis lived alongside many terrifying predators like crocodiles, big cats, and hyenas, with their prowess in tools proving to be a great evolutionary advantage. Traditionally considered the first toolmakers, Homo habilis created the Oldowan stone tool industry mainly to butcher animals. These tools were a major development in terms of technology and gave Homo habilis more options and versatility in discovering food choices. Early Homo are generally thought to have eaten a lot of meat, and in the case of Homo habilis, scavenged meat. Meat eating gave rise to a smaller gut and also provided the nutrition that gave them a larger brain of 37 to 49 cubic inches, or 600 to 800 cubic centimeters. Early hominins were typically interpreted as having lived in polygynous societies, or part of societies in which male individuals had more than one female mate at a time, though this is highly speculative, assuming Homo habilis society was similar to that of modern savanna chimps and baboons. Groups may have included 70 to 85 members, with multiple males to defend against open savanna predators like big cats, hyenas, and crocodiles. Homo habilis coexisted with other hominins like Homo rudolfensis, Homo ergaster or Homo erectus, and Paranthropus boisei. To understand more about the anatomy and culture of Homo habilis, we first need to learn about the taxonomy, research history, and naming of Homo habilis. Olduvai Gorge, Tanzania. Jonathan and Mary Leakey are seeking to uncover hominin fossils. For context, the Leakey family includes notable archaeologists like Lewis and Mary Leakey that have significantly contributed to our understanding of early hominins. Lewis and Mary Leakey are Jonathan Leakey's parents. Today, November 4, 1960, is their lucky day, with their work leading to the discovery of OH or Olduvai hominid 7, dubbed Johnny's Child. This find, including a partial juvenile skull, hand, and foot bones, dates to 1.75 million years ago. Nonetheless, it turns out that the first actual remains, OH4 a molar, were discovered by the senior assistant of Lewis and Mary Leakey, Haslan Mukiri, in 1959, yet this was not realized at the time. By this time, Lewis and Mary had spent decades, roughly 29 years, since the 1930s, excavating in Olduvai Gorge for early hominin remains, but had instead recovered mainly other animal remains in addition to the Oldowan stone tool industry. This proved to be an exciting discovery, as they did not know who made the Oldowan stone tool industry, something that remained a mystery for decades. The industry had been ascribed to Paranthropus boisei, at the time Synyanthropus in 1959, as it was the first and only hominin recovered in the area, but following the discovery of Johnny's Child, Olduvai hominid 7, we finally unraveled the secret. In 1964, Lewis Leakey, South African paleoanthropologist Philip B. Tobias, and British primatologist John R. Napier officially assigned the remains into the genus Homo, as recommended by Australian anthropologist Raymond Dart. Homo habilis means able, handy, or mentally skillful, and vigorous in Latin. The specimen's association with the Oldowan, then considered evidence of advanced cognitive ability, was also part of the reason for classifying it into Homo. The other reasons for classification included a human-like foot, the increased cranial capacity and comparatively smaller premolar and molar teeth of the fossils, and hand bones that suggested an ability to manipulate objects with precision. These traits are all characteristic of early human species, hence the naming. This led to the species being dubbed Homo habilis, or handyman. Olduvai hominid 7 was designated as the Hall-type specimen. Homo habilis were the earliest of our ancestors to show a significant increase in brain size and also the first to be found associated with stone tools. How these two aspects are related are some of the topics paleoanthropologists ponder and study. It was the year 1986. Homo habilis skeletal anatomy was unclear, yet this year would mark the discovery of Olduvai hominid 62, a fragmentary skeleton discovered by American anthropologist Tim D. White in association with Homo habilis skull fragments that would definitively establish aspects of Homo habilis skeletal anatomy and reveal more australopithecus-like than Homo-like features. Because of this, as well as similarities in dental adaptations, paleoanthropologist Bernard Wood and biological anthropologist Mark Collard suggested moving the species to Australopithecus in 1999. Associations with more ape-like Australopithecine traits or human-like Homo traits has been the subject of debate amongst paleoanthropologists in regards to Homo habilis. This means that the re-evaluation of Olduvai hominid 62 to a more human-like physiology, if correct, would cast doubt on the claim supporting Homo habilis being more Australopithecine-like. These two opposing fields of evidence challenge the classification of Homo habilis. The discovery of the 1.8 million year old Georgian Damani sea skulls in the early 2000s, which exhibit numerous similarities with early Homo, has led to paleoanthropologists suggesting that all contemporary groups of early Homo in Africa, including Homo habilis and Homo rudolfensis, are the same species and should be assigned to Homo erectus, which is certainly a controversial claim we need to examine more closely. Following our journey into Homo habilis taxonomy, we will now examine its anatomy. It has generally been thought that brain size increased along the human line especially rapidly at the transition between species, with Homo habilis brain size being smaller than that of Homo ergaster or Homo erectus, jumping from about 37 to 40 cubic inches or 600 to 650 cubic centimeters in Homo habilis to about 55 to 61 cubic inches or 900 to 1000 cubic centimeters in Homo ergaster or Homo erectus. Nonetheless, a 2015 study showed that the brain sizes of Homo habilis, Homo rudolfensis, and Homo ergaster generally range between 31 to 55 cubic inches or 500 to 900 cubic centimeters after reappraising the brain volume of OH7 from 39.5 to 41.9 cubic inches or 647 to 687 cubic centimeters to 44.5 to 50.3 cubic inches or 729 to 824 cubic centimeters. This indicates a jump from Australopithecine brain size, which generally ranged from 24 to 31 cubic inches or 400 to 500 cubic centimeters. Here is a diagram about the increase in hominin cranial capacity over time. The brain anatomy of all Homo features an expanded cerebrum in comparison to Australopithecines. This led to physiological changes that would lead to vastly different lifestyles and skills. The pattern of striations on the teeth of OH65 slanting right, which may have been accidentally self-inflicted when the individual was pulling a piece of meat with its teeth, and the left hand while trying to cut it with a stone tool using the right hand. If correct, this could indicate right-handedness. And handedness is associated with major reorganization of the brain and the lateralization of brain function between the left and right hemispheres. Lateralization of brain function means that specific mental processes are mainly specialized to one side or the other. This scenario has also been hypothesized for some Neanderthal specimens. Lateralization could be implicated in tool use. In modern humans, lateralization is weakly associated with language. Understanding the lateralization of the brain function of early Homo fossils may unravel mysteries about human evolution. Here is the average capacity of the brain case in fossil hominins. Homo habilis was the first species to exhibit enlarged Broca's and Wernicke's areas. The Broca area is also known as the motor speech area, regulating breathing patterns while speaking and vocalizations necessary for normal speech. On the other hand, the Wernicke area is a region of the brain that includes motor neurons involved in the comprehension of speech. This means they may have had the motor control that allowed more lingual or activity related to the tongue, and the ability to comprehend the resulting sounds they could produce. This suggests a proto-language or a method of communication that could have led to different outcomes compared to Australopithecine ancestors. The tooth rows of Homo habilis were B-shaped as opposed to U-shaped in later Homo, and the mouth jutted outwards, meaning it was prognathic, though the face was flat from the nose up. This is one of the differences between Australopithecines, a flatter face and shorter tooth row. Nonetheless, the postcranial anatomy of Homo habilis is still reminiscent of apes in terms of relative proportions, How so, you may ask? Well, let us now delve into its anatomical build. Based on the fragmentary skeletons OH62, presumed to be a female, and KNM-ER 3735, presumed to be a male, Homo habilis' body anatomy has generally been considered to have been more ape-like than even that of the earlier Australopithecus afarensis, and consistent with an at least partially arboreal lifestyle in the trees as is assumed in australopithecines. Based on OH62, and assuming comparable body dimensions to australopithecines, Homo habilis has generally been interpreted as having been small-bodied like australopithecines, with OH62 generally estimated at about 3 feet 3 inches to 3 feet 11 inches, or 100 to 120 centimeters in height, and 44 to 82 pounds, or 20 to 37 kilograms in weight. However, assuming longer, modern human-like legs, OH62 would have been about 4 feet 10 inches, or 148 centimeters, and 77 pounds, or 35 kilograms, and KNM-ER 3735 about the same size. Paleoanthropologists have different estimates based on two assumptions. First, australopithecine-like proportions, and second, human-like proportions. These contrasting estimates reveal insights into evolutionary relationships. For comparison, modern human men and women in the year 1900 averaged 5 feet 4 inches or 163 centimeters and 5 feet or 152.7 centimeters, respectively. It is generally assumed that pre-Homo ergaster hominins, including Homo habilis, exhibited notable sexual dimorphism, with males markedly bigger than females. Nonetheless, as scientists, we have to question our assumptions and seek answers without bias. Additionally, relative female body mass is unknown in this species. Early hominins, including Homo habilis, are thought to have had thick body hair coverage like modern non-human apes because they appear to have inhabited cooler regions and are thought to have had a less active lifestyle than presumed hairless post-ergaster species. Evolutionary pressures shaped early Homo species through diverse pathways. Consequently, they probably required thick body hair to stay warm. This inference shows how paleoanthropologists and other scientists employ inductive and deductive reasoning when drawing conclusions and speculating. Based on dental development rates, Homo habilis is assumed to have had an accelerated growth rate compared to modern humans, more like that of modern non-human apes. This means that their growth rate is more ape-like than human-like. Homo habilis is an incredible example of a hominin that exhibits transitional traits between ape-like ancestors and the later Homo. The arms of Homo habilis and australopithecines have generally been considered to have been proportionally long and thus adapted for climbing and swinging, possibly representing adaptations for an arboreal lifestyle. In 2004, anthropologists Martin Hausler and Henry McHenry argued that because the humerus to femur ratio of OH62 is within the range of variation for modern humans, and KNM-ER 3735 is close to the modern human average, it is unsafe to assume ape-like proportions. However, we have to view the evidence. The humerus of OH62 measured 10.2 to 10.6 inches, or 258 to 270 millimeters long, and the ulna or forearm 9.6 to 10 inches, or 245 to 255 millimeters, which is closer to the proportions seen in chimps. The hand bones of OH7 suggest precision gripping, important in dexterity, as well as adaptations for climbing. These finds show the mosaic-like distribution of ape and Homo-like traits. To reconstruct our fragmented past, evolutionary relationships must be analyzed and compared. In regard to the femur, traditionally, comparisons with the Australopithecus afarensis specimen AL288-1 have been used to reconstruct stout legs for Homo habilis, but Hausler and McHenry suggested the more gracile OH24 femur either belonging to Homo ergaster or Homo erectus or Paranthropus boisei may be a more suitable comparison. In this instance, Homo habilis would have had longer, human-like legs and have been effective long-distance travelers, as is assumed to have been the case in Homo ergaster. However, estimating the unpreserved length of a fossil is highly problematic, variable, and may yield inaccurate estimates. The thickness of the limb bones in OH62 is more similar to chimps, ape-like in anatomy, than Homo ergaster or Homo erectus and modern humans, which may indicate different load-bearing capabilities more suitable for arboreality in Homo habilis. The load distribution between the limbs was significantly different in Homo habilis from that of Homo erectus and modern humans, meaning these are anatomically less developed areas given the evolutionary relationships of early Homo. The strong fibula of OH35, though this may belong to Paranthropus boisei, is more ape-like and consistent with arboreality and vertical climbing. Providing generalized facts for early hominins is challenging, given the specific variation between fossil finds. OH8, a foot, is better suited for terrestrial movement than the foot of Australopithecus afarensis, though it still retains many ape-like features consistent with climbing. However, the foot has projected toe bone and compacted midfoot joint structures, which restrict rotation between the foot and ankle as well as at the front foot. Foot stability enhances the efficiency of force transfer between the leg and the foot, and vice versa, and is implicated in the plantar arch elastic spring mechanism, which generates energy while running but not walking. This could possibly indicate Homo habilis was capable of some degree of endurance running, which is typically thought to have evolved later in Homo ergaster or Homo erectus. If true, odds are that a currently accepted paradigm of Homo habilis behavior would change significantly. As a review of primitive ape-like characteristics, Homo habilis exhibited some degree of prognathism, large incisors, curved phalanges, long arms and short legs, and thick metatarsals. As a review of derived traits, Homo habilis exhibited gracile craniofaciodental traits, like a thin skull bald, a more globular cranium, an expanded frontal lobe, possibly left hemispheric dominance, and large Broca's and Wernicke's areas in the brain, a reduced supraorbital torus, a smaller mandible, canines and cheek teeth, a parabolic dental arcade, increased manual dexterity, a large femoral head and acetabulum, and a shorter neck, and finally a more stable foot with shorter toes, and exhibiting a loss of the ape-like divergent hallux or big toe. Bipedalism and the growth of a larger brain were two major steps in the process of changing the ape-like ancestor into modern humans. With larger brains, the beginnings of speech, and tools to aid meat eating, Homo habilis and its pre-human branches left the first traces of cultural behavior. Typically, Homo ergaster or Homo erectus is considered to have been the first human to have lived in a monogamous society, or part of a society in which individuals had only one mate at a time, and all preceding hominins were polygynous, or part of societies in which male individuals had more than one female mate at a time. However, it is highly difficult to speculate with any confidence the group dynamics of early hominins. The degree of sexual dimorphism and the size difference between males and females is often used to correlate between polygyny with high differences or disparity and monogamy with low differences or disparity based on general trends, though not without exceptions seen in modern primates. Rates of sexual dimorphism are difficult to determine as early hominin anatomy is fragmentary and often incomplete, making it rather challenging for paleoanthropologists to examine, and are largely based on few specimens. In some cases, sex is randomly determined in large part based on perceived size and apparent robustness in the absence of more reliable elements in sex identification such as the pelvis. Mating systems are also based on dental anatomy, but early hominins possess a mosaic anatomy of different traits not seen together in modern primates, with evolutionary pathways leading to transitional and more advanced or less advanced forms. The enlarged cheek teeth would suggest marked size-related dimorphism, and thus intense male-to-male conflict over mates and a polygynous society, but the small canines should indicate the opposite. Other selective pressures, including diet, can also significantly impact dental anatomy. The spatial, or distribution in space, of tooled and processed animal bones at the FOK Singe 22 and PTK 1 sites in Al Dubai Gorge indicate the inhabitants use this area as a communal butchering and eating ground, as opposed to the nuclear family system of modern hunter-gatherers, where the group is subdivided into smaller units, each with their own butchering and eating grounds. The behavior of early Homo, including Homo habilis, is occasionally modeled on that of savanna chimps and baboons, social primates that live in groups and cooperate with each other. These communities consist of several males, as opposed to a harem society, in which a group of females share one male partner. This is to defend the group on the dangerous and exposed habitat, sometimes engaging in group display of throwing sticks and stones against enemies and predators. Homo habilis might have employed similar strategies. The left foot OH8 seems to have been bitten off by a crocodile, possibly Crocodylus anthropophagus, and the leg OH35, which either belongs to Paranthropus boisei or Homo habilis, shows evidence of leopard predation. Life for Homo habilis was challenging, and survival was tough, often facing threats in new situations where they had to adapt to thrive. Homo habilis and contemporary hominins were likely predated upon by other large carnivores of the time, such as, in Olduvai Gorge, the hunting hyena Chasmapordithes nitidula, and the saber-toothed cats Dinofelis and Meganterion. In 1993, American paleoanthropologist Leslie C. Aiello and British evolutionary psychologist Robin Dunbar estimated that Homo habilis group size ranged from 70 to 85 members, on the upper end of chimp and baboon group size, based on trends seen in neocortex size and group size in modern apes or non-human primates. Homo habilis coexisted with Homo rudolfensis, Homo ergaster or Homo erectus, and Paranthropus boisei. Nonetheless, paleoanthropologists are unclear about how all of these species interacted. To explain why Paranthropus boisei was associated with Oldowan tools despite not being the napper or the one who made the tools, Leakey and colleagues, when describing Homo habilis, suggested that one possibility was Paranthropus boisei was killed by Homo habilis, perhaps as food. Nonetheless, when describing Paranthropus boisei 5 years earlier, Louis Leakey said, "There is no reason whatever, in this case, to believe that the skull represents the victim of a cannibalistic feast by some hypothetical more advanced type of man." Contradictory pieces of evidence puzzle paleoanthropologists seeking solutions to the situations behind many fossils. Early Homo had smaller teeth than Australopithecus, yet their tooth enamel was still thick and their jaws were still strong, suggesting their teeth were still adapted to chewing hard foods, though possibly only seasonally, when their preferred foods became less available. This is an example of adaptation to new or seasonal environmental conditions, like droughts or famine. Dental microwear studies suggest that the diet of Homo habilis was flexible and versatile, and that they were capable of eating a myriad of foods, including some tougher foods like woody plants, some animal tissues, and leaves. Yet they did not routinely consume or specialize in consuming hard foods like seeds, very hard tubers, dried meat, or brittle nuts. At this point, you might be asking, what about the bones? Well, another line of evidence for the diet of Homo habilis comes from some of the earliest percussion and cut-marked bones, discovered back to 2.6 million years ago. Scientists typically associate these traces of butchery of large animals, including direct evidence of marrow and meat eating, with the earliest appearance of the genus Homo, including Homo habilis. This marrow would be rich in nutrients and provide energy, essential for the development and survival of early Homo. Many scientists think early Homo, including Homo habilis, made and used the first stone tools found in the archaeological record, dating back to around 2.6 million years ago. Nonetheless, this hypothesis is challenging to test because several other species of early human lived at the same time, even in the same geographic region where traces of the earliest tool use have been discovered. This creates a muddle of fossils whose napper is unknown or contested, leading to ambiguities and more questions. Paleoanthropologists seek to resolve this by weighing evidence and drawing connections between fossils and species. It is thought Homo habilis derived meat from scavenging rather than hunting, which is the scavenger hypothesis, acting as a confrontational scavenger and stealing kills from smaller predators like jackals or cheetahs. Fruit was likely also an important dietary component, indicated by dental erosion consistent with repetitive exposure to acidity. Based on dental microwear texture analysis, Homo habilis, like other early Homo, likely did not regularly consume tough foods. Microwear texture complexity is, on average, somewhere between that of tough food eaters and leaf eaters or follow-bores, and points to an increasingly generalized and omnivorous diet. it is typically thought that the diets of Homo habilis and other early Homo had a greater proportion of meat than Australopithecus, and that this led to brain growth. The main hypotheses regarding this are: meat is energy and nutrient rich, and puts evolutionary pressure on developing enhanced cognitive skills to facilitate strategic scavenging and monopolize fresh carcasses. In other words, meat allowed the large and calorie-expensive ape gut to decrease in size, allowing this energy to be diverted to brain growth. Alternatively, it is also suggested that early Homo, in a drying climate with scarcer food options, relied primarily on underground storage organs like tubers and food sharing, which facilitated social bonding among both male and female group members. This flexibility might have in fact been the source of Homo habilis' success as a species that thrived for hundreds of thousands of years. However, unlike what is presumed for Homo ergaster and later Homo, short-statured early Homo are generally considered to have been incapable of endurance running and hunting, and the long and australopithecus-like forearm of Homo habilis could indicate early Homo were still arboreal to a degree. This would create a disruption in our paradigm of Homo habilis as possessing more primitive ape-like traits than human-like traits, and may even elicit a new classification. Additionally, organized hunting and gathering is thought to have emerged in Homo ergaster. Nonetheless, the proposed food-gathering models to explain large brain growth necessitate increased daily travel distance. It has also been argued that Homo habilis instead had long, modern human-like legs and was fully capable of effective long-distance travel, while still remaining at least partially arboreal, or dwelling in trees, which would be a midpoint between the two opposing fields of evidence. Large incisor size in Homo habilis, compared to Australopithecus predecessors, implies this species relied on incisors more. These sharp teeth would be used to bite into food and cut it into smaller pieces as part of digestion. The bodies of the mandibles of Homo habilis and other early Homo are thicker than those of modern humans and all living apes, more comparable to Australopithecus. The mandibular body resists torsion from the bite force or chewing, meaning their jaws could produce unusually powerful stresses while eating. This would imply a dietary pathway. The greater molar cusp relief in Homo habilis compared to Australopithecus suggests that Homo habilis used tools to fracture tough foods, like pliable plant parts or meat. Otherwise, the cusp would have been more worn down. Molar cusp relief is the relative distance between cusp height and intercusp valleys. Nonetheless, the jaw adaptations for processing mechanically challenging food indicates technological advancement did not greatly affect diet. This means other changes were responsible for evolutionary development. Homo habilis is associated with the early Stone Age Oldowan stone tool industry, found at the Oldowan industrial complex area. These early tools were usually made with one or a few flakes chipped off with another stone. It was followed by the more sophisticated Aquilon industry. Scientists are currently investigating the Oldowan industrial complex area in East Africa to uncover details about the development of technology by early hominins. It turns out that individuals likely used these tools primarily to butcher and skin animals, crush bones, but also sometimes to saw and scrape wood and cut soft plants. Knappers, or the creators of the tools, appear to have carefully selected lithic cores and knew that certain rocks would break in a specific way when struck hard enough and on the right spot, and they produced diverse types, including choppers, polyhedrons, and discoids. The tools are made of pebbles of quartz, quartzite, or basalt, and are chipped in two directions to form simple, rough, all-purpose tools capable of cutting, scraping, or chopping. Flakes remaining from tool development were also used as tools. Nonetheless, specific shapes were likely not thought of in advance, and odds are that they are due to a lack of standardization in producing such tools, as well as the types of raw materials at the knappers' disposal, which would have varied from area to area. For instance, spheroids are common at Al Dubai, which features an abundance of large and soft quartz and quartzite pieces, whereas Cubiphora lacks spheroids and provides predominantly hard basalt lava rocks. Environmental conditions elicit new solutions to daily challenges faced by Homo habilis and early Homo species. Until the later Aquilon culture invented by Homo ergaster or Homo erectus, Oldowan technology does not require planning and foresight to manufacture. and thus does not indicate high cognition in Oldowan nappers, though it does require a degree of coordination and some knowledge of mechanics. Acheulean culture was distinguished by oval and pear-shaped hand axes associated with Homo erectus and derived species like Homo heidelbergensis. Acheulean tools arose from the earlier Oldowan technology associated with Homo habilis. Oldowan tools infrequently exhibit retouching and were probably discarded immediately after use most of the time. If true, this means they were ephemeral tools. The Oldowan was first reported in 1934, but it was not until the 1960s that it became widely accepted as the earliest culture, dating to 1.8 million years ago, and as having been manufactured by Homo habilis, among the first tool makers in the human lineage. These tools were first found with the Homo habilis remains in Oldobai Gorge, a site known for yielding a rich collection of early hominin fossils, and subsequently in other areas of eastern, central, and southern Africa, making the Oldowan stone tool industry the earliest evidence we have of human cultural behavior. From this, paleoanthropologists were able to more accurately confirm and deduce the range of Homo habilis. Since then, more discoveries have placed the origins of material culture substantially backwards in time, with the Oldowan being discovered in Ledijararu and Gona in Ethiopia, dating to 2.6 million years ago, perhaps associated with the evolution of the genus. But we should not disregard earlier hominins as not crafting tools, as australopithecines are known to have manufactured tools, like the 3.3 million year old Lomekwi stone tool industry, and some evidence of butchering from about 3.4 million years ago. However, the degree of complexity and development in the tools these two groups made, Homo habilis and Australopithecus, is significant. The comparatively sharp-edged Oldowan culture was a major innovation from Australopithecine technology, and it would have allowed different feeding strategies and the ability to process a wider range of foods, which would have been advantageous in the changing climate of the time. It is unclear if the Oldowan was independently invented or if it was the result of hominin experimentation with rocks over hundreds of thousands of years across multiple species. Unraveling this mystery would define a clear picture of the origin of stone tools in early hominins. The current evidence points to tool making and meat eating occurring by 3.3 million years ago, yet only a handful of sites with stone tools and or butchered animal bones have been found before roughly 1.8 million years ago. The earliest site with evidence that early humans repeatedly returned to one place to make stone tools and butcher animals, a site in Kenya known as Kengera South, is dated to 2 million years ago. This seems to be the beginning of consistent butchery activities. The site of Kengera occurs on the Homa Peninsula of western Kenya, located in an area surrounded by eroded volcanic edifices, on the southern shore of the Winam Gulf of Lake Victoria. The Smithsonian Institution has been excavating the site for decades, yielding valuable discoveries. The following sites contain evidence of stone tools and their creation. First, Lomekwi 3, Lake Turkana region, Kenya, dating to 3.3 million years ago, yielded cores, anvils, and flakes. Second, a site in Gona, Ethiopia, greater than 2.5 million years ago, yielding 3,000 stone artifacts. Third, a site in Hadar, Ethiopia, dating to 2.4 million years ago, with tools discovered in addition to a Homo habilis mandible. Fourth, one of the richest of all, Oldobai Gorge, Tanzania, dating to 1.8 million years ago, yielding numerous tools. Finally, Kubifora, Kenya, yielding a high concentration of flakes suggesting repeated use. Toolmaking would have facilitated access to a wider range of foods and the ability to process these foods more intensively or efficiently, likely making them more palatable and yielding more calories. In the case of meat and marrow eating, toolmaking would have opened up new sources of food higher in protein, fat, and calories than many other foods available in African savanna landscapes. The year was 1962. An intriguing structure was uncovered that puzzled scientists. It was a 12 feet by 14 feet by 1 feet, or 366 centimeter by 427 centimeter by 30 centimeter long circle made with volcanic rocks discovered in Oldobai Gorge, at 2 to 2.5 feet or 61 to 76 centimeter intervals. Rocks were piled up to 6 to 9 inches or 15 to 23 centimeters high. But why was this made? What purpose did it serve? Well, British paleoanthropologist Mary Leakey suggested the rock piles were used to support poles stuck into the ground, possibly to support a windbreak or a roth hut. This discovery shifted our paradigm of what early hominins could build. Some modern-day nomadic tribes built similar low-lying rock walls to build temporary shelters upon, bending upright branches as poles and using grasses or animal hide as a screen. Dating to 1.75 million years ago, it is attributed to some early Homo, and is the oldest claimed evidence of architecture. Homo habilis was named by Lewis and Mary Leakey and colleagues in 1964. Synonyms include Australopithecus habilis, named by Wood and Collard in 1999, Homo microcranus, named by Ferguson in 1995, Homo gottingensis, named by Curnow in 2010, and Homo rudolfensis, named by Alexeyev in 1986. There is still no wide consensus as to whether or not Homo habilis is ancestral to Homo ergaster or Homo erectus, or is an offshoot, meaning that it developed from the human line, and whether or not all the specimens assigned to Homo habilis are correctly assigned or the species is an assemblage of different Australopithecus and Homo species. If true, this would shift the whole taxonomy and classification of Homo habilis and may possibly be identified as a wastebasket taxon. Nonetheless, Homo habilis and Homo rudolfensis generally are recognized members of the genus at the base of the family tree, with arguments for synonymization or removal from the genus not widely adopted. This, however, may change over time with more discoveries and insights leading to better informed phylogeny and classification. Homo habilis, along with Homo rudolfensis, is one of the earliest members of the genus Homo. Many scientists think it is an ancestor of later species of Homo, possibly on our own branch of the family tree. Naming this species required a redefining of the genus Homo, including reducing the lower limit of brain size, sparking a heated debate about the validity of this species. The current debates about Homo habilis are mostly about the amount of species of Homo habilis and whether Homo habilis should be classified as Homo or Australopithecus. Here is the Homo family tree showing Homo habilis and Homo rudolfensis at the base as offshoots of the human line. Though it is now largely agreed upon that Homo evolved from Australopithecus, the timing and placement of this split has been much debated, with many Australopithecus species having been proposed as the ancestor. Nonetheless, we need to look at fossil evidence to understand these nuances. The discovery of LD351, the oldest Homo specimen, dating to 2.8 million years ago in the Afar region of Ethiopia, may indicate that the genus evolved from Australopithecus afarensis around this time. The species LD351 belongs to could be the ancestor of Homo habilis, but this is unclear. The oldest Homo habilis specimen, AL6661, dates to 2.3 million years ago, but is anatomically more derived, meaning it has less ancestral or basal, meaning primitive, traits than the younger OH7, suggesting derived and basal or primitive morphs live concurrently, and that the Homo habilis lineage began before 2.3 million years ago. Based on 2.1 million year old stone tools from Shangshan, China, Homo habilis or an ancestral species may have dispersed across Asia. The youngest Homo habilis specimen, OH13, dates to about 1.65 million years ago. Here is the African hominin timeline in millions of years ago. Well, what about Australopithecus remains? What do these fossils tell us about our ancient ancestors? While the origin of Homo habilis has been in a state of flux in recent years, the discovery of Australopithecus sediba has raised more questions about the origin of our genus. The discovery of Lucy in the early 1970s led some researchers to turn away from Australopithecus africanus in favor of Australopithecus afarensis as the ancestor of the genus Homo. In recent years, the idea that a cladistic event had occurred with Australopithecus afarensis, leading to Australopithecus africanus and the more derived and robust forms, on the one hand, and the genus Homo on the other, gained popularity. Australopithecus sediba now seems to have bridged the gap between the Australopiths and the genus Homo, sharing traits with Australopithecus africanus, Homo habilis, and Homo ergaster. The similarities with the two Homo species may help resolve the problem as to which of the two species of early Homo gave rise to Homo ergaster. There are proponents in support of each of the evolutionary scenarios, with their share of pros and cons. Now, what about its scientific classification? Following description, it was intensely debated if Homo habilis should be reclassified into Australopithecus africanus, the only other early hominin known at the time, mainly because the remains were so old, and at the time Homo was presumed to have evolved in Asia, with the Australopithecines having no living descendants. This view was modified with additional discoveries. Moreover, the brain size was smaller than what Wilfrid Le Gros Clark proposed in 1955 when considering Homo, leading to another change in our paradigm of the anatomy of early Homo. The classification as Homo habilis began to receive wider acceptance as more fossil elements and species were unearthed. In 1983, South African paleoanthropologist Philip Tobias proposed that Australopithecus africanus was a direct ancestor of Paranthropus and Homo, with the two being sister taxa, and that Australopithecus africanus evolved into Homo habilis, which evolved into Homo erectus, which evolved into modern humans, by a process of cladogenesis. Cladogenesis is essentially when a parent species splits into two distinct species. He further stated that there was a significant evolutionary leap between Australopithecus africanus and Homo habilis, and that human evolution progressed gradually because Homo habilis' brain size had nearly doubled compared to Australopithecine predecessors. Here is human evolution according to Tobias in 1983. For the longest time, many had accepted Tobias's model and assigned late Pliocene to early Pleistocene hominin remains outside the range of Paranthropus and Homo erectus into Homo habilis. For non-skull elements, this was done on the basis of size, as there was a lack of clear observed traits to define a particular species. Because of these practices, there was an ample range of variation, and the terms Homo habilis sensu stricto, or in the strict sense, and Homo habilis sensu lato, or in the broad sense, were in use to include and exclude, respectively, more divergent, different unique morphs. To address this, in 1985, English paleoanthropologist Bernard Wood proposed that a comparatively massive skull, KNM-ER 1470 from Lake Turkana, Kenya, discovered in 1972 and assigned to Homo habilis, actually represented a different species, now referred to as Homo rhodolfensis. It is also argued that instead, it represents a male specimen, whereas other Homo habilis specimens are female. Connecting these pieces of evidence has been challenging for paleoanthropologists. Early Homo from South Africa have variously been assigned to Homo habilis, or Homo ergaster or Homo erectus, yet designation has largely been unclear. In 2010, Australian archaeologist Darren Carroll proposed splitting off South African early Homo into a new species, Homo godingensis. Homo habilis belongs to the kingdom Animalia, the phylum Cridata, the class Mammalia, the order Primates, the suborder Haplorhini, the infraorder Simiforms, the family Hominidae, the subfamily Homininae, the tribe Hominini, the genus Homo, and the species Homo habilis. Here is a summary of Homo habilis facts. Here is a table of key Homo habilis fossil locations and the corresponding fossils and dates. Fossil representation includes numerous remains, with some possibly attributed to other species, such as KNM-BC 1, KNM-ER 1470, 1805, 1813, OH7, 8, 13, 16, 24, 35, 62, 65, and SKA 47. I'll be going over each of these to understand what discoveries these fossils have yielded. These remains, consisting of a right temporal bone or ear region, were discovered by John Kimingich in 1965 and a team led by Andrew Hill in the site Chemeron, Kenya, dating to about 2.4 million years ago. Some paleoanthropologists have attributed the fossil to Paranthropus or Australopithecus. Analysis led to the ruling out of Homo ergaster or Homo erectus, Paranthropus, and Australopithecus as possibly representing this find, considering the fossil to be compatible with traits of Homo habilis and Homo rhodolfensis, or an early transitional species of Homo. The specimen KNM-ER 1470 was found in 1972 at Kubifora, Kenya, by a team under Richard Leakey. Dated at close to 2 million years, This specimen is classified by some paleoanthropologists as Homo rudolfensis, though its classification is highly controversial. Some classify it as Homo habilis, while others as Homo rudolfensis. This is because it resembles both Australopithecus and Homo, and is highly fragmentary. It had a brain volume of about 46 cubic inches or 750 cubic centimeters. This fossil was discovered by Paul Abel in the site Kubifora, Kenya in 1974, dating to about 1.7 million years ago. KNM-ER 1805 was found divided into three main parts: a cranium, a maxilla, and a mandible. The individual was an adult at the time of death, as evidenced by the third molars. Debate as to the correct classification of this specimen has existed ever since it was discovered. It was first placed in Homo erectus, yet based on the degree of prognathism and the shape of the cranium, especially the prominent nuchal crest, it is now considered to belong in Homo habilis. KNM-ER 1813 was discovered by Kamoja Kimu in the site Kubifora, Kenya in 1973, with the remains being dated as about 1.9 million years old. This fossil is one of the most complete skulls of this species, possibly making it the best known Homo habilis specimen. Known from the Turkana Basin and Olduvai Gorge in East Africa, it had an overall smaller size than other Homo habilis fossils, yet with a fully adult and typical Homo habilis morphology. Its cranial capacity was estimated at only 31 cubic inches or 510 cubic centimeters. The lower jaw of OH7, a specimen found in 1960 at Olduvai Gorge, Tanzania, by Jonathan and Mary Leakey, was identified by Louis Leakey and others in 1964 as a fossil of Homo habilis and made the holotype specimen. It was dubbed Johnny's Child. The remains date to 1.75 million years ago and consist of fragmented parts of a lower mandible, an isolated maxillary molar, 2 parietal bones, and 21 finger, hand, and wrist bones. The hand possesses human and ape-like traits. These, among other anatomical features, defined it as a transitional species between Australopithecus africanus and Homo erectus, leading to debate regarding classification. The cranial capacity of this specimen was placed at 40 cubic inches or 663 cubic centimeters, based on the fact that the fossils belong to a 12 or 13-year-old male. This specimen was discovered by a team led by Louis Leakey in the site Olduvai Gorge, Tanzania in 1960, with the remains being dated as about 1.8 million years old. It is a fossilized foot exhibiting both ape and human characteristics, refuting the hypothetical prehuman foot prediction by Morton in 1935, who had predicted an atavistic divergent first ray and a very substantial robust calcaneus. The subject may have been drinking from the lake at the wrong time, when a crocodile may have sprung from the water and viciously attacked. Scientists deduced this due to tooth marks on the ankle bone, similar in shape and pattern to those made by modern crocodiles. OH13 was discovered by Ndibo Mbwika in 1963 at Olduvai Gorge, Tanzania, dating to 1.65 million years ago. It was described by Louis and Mary Leakey in 1964 and consists of a partial cranial fossil dubbed Cinderella or Cindy. The remains include 2 maxillary fragments with several teeth, part of the jaw, and parts of the skull. Its cranial capacity has been estimated at 30 cubic inches or 500 centimeters. Based on comparisons with other specimens, it would correspond to an early Homo habilis, with the variation thought by some scientists to represent sexual dimorphism. This specimen was discovered by Michael Mutumbu in 1963 in the site Olduvai Gorge, Tanzania, dating to about 1.7 million years ago. This specimen was dubbed George. This crushed skull was challenging to reconstruct and has a lightly built brain case, yet a larger brain size than Australopithecus. 2 features that led Louis Leakey and colleagues to recognize the species Homo habilis. It corresponds to a subadult, with its brain having a volume of 38 cubic inches or 623 cubic centimeters. Like OH13, it was found associated with stone tools. This specimen, dubbed Twiggy, was discovered by Peter Nazub in the site Olduvai Gorge, Tanzania in 1968, dated as about 1.8 million years old. OH24 is the oldest fossil skull discovered at Olduvai Gorge, and besides OH5, it is the most complete. The skull was found crushed almost flat, and was therefore named after the famously skinny model of the time, Twiggy. It represents an adult individual which died soon after the eruption of its third molars. Its face is prognathic, or projecting forward under the nose, as in other Homo habilis fossils, yet unlike Australopithecus. It also exhibits a larger cranial bald, indicating an expansion in brain size from its ancestor, and a reduction of facial prognathism, or the size of the face typical of the evolution of early Homo. OH35 includes a Homo habilis tibia and fibula found in Olduvai Gorge, Tanzania, in 1960 by Louis Leakey's assistants, and dated at 1.8 million years. The publication of this finding was carried out by Mary Leakey in 1971. Though these remains may belong to Paranthropus boisei, they are more ape-like and consistent with arboreality and vertical climbing. They may come from the same individual as other fossils including OH8, a foot, or OH10, a terminal toe phalanx. These leg bones show evidence of leopard predation. This specimen, discovered by Tim D. White in Olduvai Gorge, Tanzania in 1986, is dated to 1.8 million years ago. It consists of a jaw with teeth and skull fragments as well as pieces of a right arm and both legs. These remains establish aspects of Homo habilis skeletal anatomy for the first time, and reveal that Homo habilis had more Australopithecus-like than Homo-like features. The skeleton may have belonged to a Homo habilis female, made up of more than 300 fragments that allowed for this observation. Nonetheless, this does not mean it did not possess traits similar to Homo. The largest remains are the maxilla and parts of the long bones, yet reconstructing their length is challenging. This has led to controversy between bone proportions and consequently diverse classifications. This jaw, with complete dentition, is part of a fossil skull of Homo habilis, with an estimated age of 1.8 million years in the Calabrian of the Pleistocene, discovered in 1995 by Amy Cushing and Agostino Benanz in Olduvai Gorge and first described by R.J. Blumenschein and colleagues in 2003. It is preserved in the National Natural History Museum in Arusha, Tanzania. It shows features of Homo habilis and resembles KNM-ER 1470, the holotype of Homo rudolfensis. The molar crowns are small relative to the length of the roots, and the roots of the canines are long and narrow. The palate resembles KNM-ER 1470. Due to these similarities, it has been suggested that Homo habilis and Homo rudolfensis be unified into one species. From the dental marks produced by the tools used in eating, such as cutting food, it can be concluded that the individual was right-handed. This specimen was discovered by Ronald Clark in the site Swartkrans, Republic of South Africa, in 1969, dating between 1.8 and 1.5 million years ago. This fossil has similarities to early African Homo erectus, sometimes known as Homo ergaster, yet it shows other similarities to Homo habilis, also known to occur from eastern or southern Africa. Due to how it was a composite skull, reassembled from 3 separate pieces, there's been debate as to its exact affinities. It has traits suggesting it belongs to the genus Homo, yet also evidence for Australopithecine affinities, or even Homo godengensis, a new species proposed by Darren Curnow. Homo habilis has appeared in popular culture in the 2003 French television show A Species Odyssey as a hairy hominin that arose due to environmental conditions like a colder climate. It is claimed that its larger brain leads to more complex thoughts and greater intelligence. In the episode, it is shown scavenging a crocodile carcass and crafting some of the earliest stone tools. It also appeared in a 2019 episode of Japanese public media organization NHK, known as Dawn of the Stone Age, Out of the Cradle, confronting a pack of hyenas to steal a carcass and forging the first stone tools. Moreover, it is depicted in the 2003 Walking with Cavemen episode 2, Blood Brothers, as inquisitive scavengers fighting off other hominin competition, yet this group faces challenges due to the loss of their leader at the jaws of a lion. We still do not know everything about our early ancestors. with paleoanthropologists continually in the field, using innovative technology, excavating new regions, and filling in some of the knowledge gaps of human evolution. Some of the unanswered questions about Homo habilis that may be answered with future discoveries include: first, was Homo habilis on the evolutionary lineage that evolved until later species of Homo and even perhaps our species Homo sapiens? Second, are Homo habilis and Homo rudolfensis indeed different species, or are they synonyms, even part of a single variable species? Or were they distinct, and one was the ancestor of the other? Third, if Homo habilis is not the ancestor of Homo erectus, how does it fit into our evolutionary tree? Fourth, Homo habilis is one of the earliest members of the genus Homo. Was there a relationship between the origin of this genus and climate change, either with an increased period of climatic fluctuations or major episodes of global cooling and drying leading to the spread of C4 grasslands? These questions prove to be challenging to answer, each possessing nuances and affected by the fragmentary nature of fossils of early hominins. Bernard Wood of George Washington University says that a convincing hypothesis for the origin of Homo remains elusive, and argues that Homo habilis should be classified neither as Homo or Australopithecus, but in its own genus. A recent review of the evolution of early Homo suggests that anatomical, physiological, and behavioral traits, long held to define our genus, did not arise in a single integrated package, but instead emerged over about 1 million years in 3 distinct lineages, with some traits evolving earlier and some later. In any case, it has become clear with more evidence that the origin of our genus remains murky, and that Homo may not have been the earliest toolmaker and meat-eater in our family tree. Unraveling these mysteries would lead to insights that would change our paradigm of early human evolution. Homo habilis, considered amongst the first toolmakers in the human lineage, is commonly known as handyman. It thrived in eastern and southern Africa roughly 2.3 to 1.65 million years ago during the gelation of the early Pleistocene, sharing its environment with numerous predators that posed a significant threat. Following a prominent description, it was the source of numerous controversies, including ideas related to synonymy, evolution, and species identification. Like contemporary Homo, its brain ranged from 31 to 55 cubic inches, or 500 to 900 cubic centimeters. A presumed female specimen of Homo habilis was interpreted as having been 3 feet 3 inches to 3 feet 11 inches, or 100 to 120 centimeters in height and 44 to 82 pounds, or 20 to 37 kilograms in weight, assuming australopithecine proportions. Yet assuming human-like proportions, it would have been almost a foot taller at 4 feet 10 inches, or 148 centimeters and 77 pounds, or 35 kilograms. Reconstructions of early hominins include thick hair and marked sexual dimorphism as adaptations to evolutionary pressures. As the creator of the Oldowan stone tools, it mainly used these for butchering. In the case of Homo habilis, it has been proposed to have been a scavenger, though this has been debated. Homo habilis has also been interpreted as having lived in polygynous societies, with males having more than one female partner at the same time. However, this is highly speculative. It may have lived in groups numbering 70 to 85 members, with multiple males to defend against open savanna predators like hyenas, crocodiles, and big cats. Homo rudolfensis, Paranthropus boise, and Homo ergaster or Homo erectus coexisted with Homo habilis. An avid toolmaker, creator of the Oldowan stone tool industry, a crucial link in human evolution, sharing Homo and australopithecine-like traits, a controversial hominin, one whose origins continue to puzzle paleoanthropologists, and an early primeval human, Homo habilis, skilled, possibly arboreal, possessing an expanded cerebrum, and a vigorous early human essential to understanding mysteries about our origins. And with that, thank you so much for joining me in this extensive primeval human episode. I certainly learned a lot about ancient human species during research. This video was a lot of fun to make. As always, thank you so much for watching. This is Enchiridion. See you next time.